Great.
Okay, make yourselves comfortable. We're ready to go. So Tynan is our speaker today, and he graduated from the life sciences program here, and then he joined the bachelor's or the master's program with Xiaolong Yang, and he's in his second year now, and in his first year, things have been going very well. It's got a peer-reviewed publication, although I guess that's on a different story.
Yeah, not necessarily mine,
yeah.
but
So that story was something that was close to my heart. Merlin, cool name, tumor suppressor, and I guess that's mostly what goes on in Xiaolong's lab, is looking at the Hippo pathway and Merlin's involvement in that. So you got your initiation, I guess, a little bit in that, but Tynan's project has been exploring this interesting new molecule, GSDMD, that is involved in pyroptosis, and he's going to be telling us about how things have been going. So, Tynan, you have the floor.
okay. Thank you, Dr. Greer, and hello, everybody, and welcome to my second seminar talk. So today I'm going to be going over my master's project that involves developing novel bioluminescent biosensors to identify small molecule inhibitors targeting GSDMD. So I'm going to begin my talk with a brief background on the gasdermin gene family, GSDMD structure and function, its role in diseases, and current GSDMD inhibitors. Following this will be my hypothesis and aims, results, future directions for the project, and then I'll end off with a summary of the talk as well as the project significance. So starting off with the background, I'm just going to be going over a brief summary of the gasdermin family. So this family is comprised of 6 related genes, GSDM A through F, which all share homologous protein structures. The first member, identified in 1998, originally known as DFNA5 and later renamed gasdermin E, was discovered during an investigation into the genetic causes of non-syndromic hearing impairment. And soon after, gasdermins began popping up in the literature and were initially found to be expressed predominantly in the gastrointestinal tract and the skin dermis, hence the name gasdermins. And over the past 2 decades, extensive research has revealed the family being involved in various cellular processes, and this includes processes such as cell differentiation, cell proliferation, and cell death. And they've also been shown to be contributing to a variety of pathological processes, being related or being a part of inflammatory related diseases, as well as contributing to tumorigenesis. So in 2015, the mechanism of gasdermin D was finally revealed, identifying it as the principal executor of a process known as pyroptosis, which is a type of programmed cell death associated with inflammation. So this discovery of GSDMD's function has given it significant attention, and it's shaped research efforts towards understanding GSDMD's inflammatory functions, as well as its implications within various diseases. So gasdermin D shares a common structure within the gasdermin family. The first component is the N-terminal domain, responsible for pore formation. The second is the C-terminal autoinhibitory domain, which represses this N-terminal. And finally, there's a linker region which connects these 2 domains together. The linker region contains a caspase cleavage site where multiple caspases cleave at this 4 residue sequence, the FLTD. And then they cleave the protein into 2 fragments between this aspartic acid and the glycine residue. So below shows a better schematic of GSDMD structure, and it illustrates how the C-terminal domain actually represses the N-terminal at this interdomain region. Once caspase-mediated cleavage occurs, causing the N-terminal to be freed, this N-terminal can then oligomerize with others and form a pore on the cell membrane. And these pores then release cellular contents, including pro-inflammatory cytokines interleukin-1 beta and interleukin-18, which is resulting in pyroptosis. So pyroptosis is the most established pathway in which GSDMD is involved in, a type of programmed cell death characterized by the release of pro-inflammatory cytokines. So this process can be triggered by a wide range of agents, including bacteria, viruses, as well as many endogenous danger signals. And pyroptosis can occur from 2 main pathways. The first is the canonical route, which involves inflammasome-mediated activation of caspase-1, whereas, or the second is then the non-canonical route, where in which directly activates caspases using lipopolysaccharides from gram-negative bacteria. So both pathways converge on caspase-mediated cleavage of GSDMD, releasing its N-terminal domain to oligomerize and form pores within or attached to the cell membrane. So these non-selective pores result in the release of pro-inflammatory cytokines and cause an osmotic imbalance due to an ion influx, leading to cell swelling and eventual lysis, as well as induced inflammatory response. So GSDMD's role in disease can be broken up to two main groups: pyroptosis-dependent and pyroptosis-independent diseases. So starting off with the pyroptosis-dependent side, GSDMD is widely expressed throughout the body, being the most prevalent gasdermin found within the entire family. It's found in vital organs such as the liver, kidney, heart, lungs, and brain. And due to its role in inflammation, it's been associated with various inflammatory-related diseases found within these tissues, such as IBS, stroke, heart failure, Alzheimer's disease, and sepsis, among others. So now what's interesting is that GSDMD has also been associated with tumorigenesis, being labeled as both a tumor suppressor as well as an oncogene, depending on the expression levels in these different types of cancer tissues. So Derek Zhang, a previous master's student in our lab, had characterized GSDMD as an oncogene in breast cancer, linking its tumorigenic role to cell metabolism, which in this case would be a pyroptosis-independent pathway or mechanism. So he showed that endogenous GSDMD is upregulated in several breast cancer cell lines. He also showed that overexpressing GSDMD actually increased cell proliferation, whereas the knockout of GSDMD in breast cancer cell lines decreased cell proliferation. He also found multiple proteins that bind with GSDMD that are involved with cell metabolism, most notably the glucose transporter GLUT1, as well as a lipid transporter SLC27A4. So the main takeaway here is that GSDMD kind of has this dualistic function in relation to its involvement with diseases. So on one hand, you have its involvement with inflammatory-related diseases, mediating pyroptosis, which cause programmed cell death and inflammation. And now, on the other hand, it's involved in tumorigenesis in ways that's not yet fully understood. So the mechanism through which it acts on tumorigenesis must involve functions that are independent of pyroptosis, since pyroptosis is a process that clearly doesn't promote cell proliferation. Instead, it likely acts on alternative mechanisms, such as cell metabolism identified by Derek, but also there might be other mechanisms that yet haven't fully been explored or discovered. So there have been multiple attempts to inhibit GSDMD activity, primarily with its involvement in pyroptosis. But unfortunately, these inhibitors don't directly target GSDMD, and instead act on upstream components in pyroptosis, such as the BAY inhibitor, which inhibits inflammasome activation, or the ZVAD-FMK inhibitor, which inhibits caspase activity. So there are also some others that act downstream, such as NSA and disulfiram, and this prevents GSDMD N-terminal oligomerization. I will say that disulfiram is the closest we've gotten to for a potential therapeutic of GSDMD, and it was a drug that was originally FDA approved to treat alcoholism. So disulfiram could conveniently modify a crucial cysteine residue on the N-terminal, and this was critical for GSDMD pore formation. But unfortunately, it did fail clinical trials due to its non-selective behavior, interacting with other cysteine residues from other components in the pathway, and this caused off-target effects. So far, there are currently no drugs that target GSDMD directly, highlighting the need to identify new candidates as potential therapeutic options. So now moving on to my hypothesis and aims of the project. Since GSDMD plays a central role in mediating multiple types of pathologies, I hypothesize identifying small molecule inhibitors that disrupt GSDMD activity will thereby prevent GSDMD-mediated diseases. In order to test this hypothesis, there's three main aims for my project. The first is developing biosensors that can detect the activity and level of GSDMD. The second is validating these biosensors and monitoring GSDMD both within living cells and in vitro. And the last is identifying and investigating small molecule inhibitors that are able to inactivate GSDMD. So moving on to the results section, I'm going to begin with the first aim with the development of the biosensors monitoring GSDMD. And in order to build this biosensor, I'll discuss nanolook binary technology, commonly referred to as nanobit for short. And it's a popular method for monitoring protein activity. So this innovative system draws inspiration from the bioluminescence produced by deep sea shrimp. The nanobit system utilizes a nanolook luciferase enzyme, which is split into two fragments termed the large bit and the small bit. And when they complement with each other, this then makes the intact nanolook enzyme. So when the large bit and small bit fragments are placed to your proteins of interest, and your proteins then bind together, this can cause a complementation of the large and small bit to make the nanolook luciferase. And then once the luciferin substrate is introduced, such as a furamazine derivative, the active luciferase enzyme catalyzes the oxidation of the substrate, resulting in the emission of bioluminescent light. So what's interesting about nanobit biosensors is in its versatility in monitoring not only the intermolecular interactions, but also the intramolecular interactions of proteins. So in my project, I've developed an intramolecular biosensor that takes advantage of GSDMD's tertiary structure, designed to track the association between the N-terminal and the C-terminal regions of the protein. So this biosensor remains active as long as full-length GSDMD is within this closed dormant state. And any external factor that disrupts this interdomain binding would cause the biosensor signal to vanish. So one way we can disrupt the biosensor signal is through GSDMD cleavage, which occurs after the various inflammatory caspases cleave it at the linker region, causing the N-terminal and the C-terminal to dissociate from each other, thereby dissociating the small and large bit. The biosensor signal can also be diminished if there's a conformational change within full-length GSDMD. So in this case, well, full-length GSDMD or GSDMD is still intact, maybe a small molecule inhibitor is preventing the N-terminal and the C-terminal from coming together, again, causing it to dissociate. And we can also monitor GSDMD levels within the cell, potentially through, or if you cause GSDMD degradation, potentially due to ubiquitination. So this gives us just a flexible and dynamic system for monitoring various aspects of GSDMD, including its stability and levels within the cell. So biosensor development begins with cloning, where the GSDMD was fused with small bit on the C-terminal and then inserted into this vector containing large bit here. And altogether, when this is transcribed and translated, you'll get an intact functional intramolecular biosensor. First, both the insert and vector undergo digestion using appropriate restriction enzymes, resulting in DNA fragments with sticky ends, which are conductive to ligation. So after this ligation takes place, the cloned vector is then introduced into the DH5 alpha competent bacterial cell line. It's a bacterial line known for producing large amounts of plasma quantities. So after the bacteria is then lysed and proteins are purified, cloning is then validated using the same restriction enzymes to digest some of the plasmid into its original components. So then these are then ran on an agarose gel, and then we try to confirm if the bands align, the bands from the vector and the insert align with its anticipated fragment sizes. So afterwards, a luciferase functional assay was conducted to assess how well this biosensor actually performs. The plasmid was transfected into the HEC293 T cells, followed by extracting the protein lysate, which would contain our biosensor. And then furamazine was introduced to gauge luciferase activity. So I want to note that an empty plasmid containing large bit alone, as well as lysate containing no plasmid, were both used to serve as controls. So essentially what this is doing is ensuring that the luminescent signal wasn't from any background activity rather than the biosensor itself. So as evident from these results here, you can see that the intramolecular biosensor does yield a robust signal, having approximately a 30-fold increase in luminescent activity compared to its control. And I also want to note too that Western blots were conducted to accompany these luciferase assays, probing for the large bit using anti-nanolook antibodies. And each sample contains the same amount of lysate. And this is basically used to confirm that the differences in luminescent activity wasn't due to differences in the amount of lysate being used. And this can be seen with the consistent beta-actin levels. So after validating luciferase complementation and successfully constructing the GSDMD intramolecular biosensor, The next step was to investigate if this complementation arises from the association between the N-terminal and the C-terminal. So to examine this, I created a mutant biosensor designed to hinder GSDMD binding, C-terminal binding to the N-terminal domain. So this mutant involves substituting 5 key residues that are located on the interdomain region of the, on the outside of the C-terminal domain and areas where this interdomain binding occurs. And these were previously shown in a paper published by Structure to be critical for this interdomain binding. So due to the lack of binding, there should be a sharp decline in signal from the mutated biosensor when comparing it to the wild type. So after site-directed mutagenesis was performed using overlapping PCR to introduce each one of these 5 mutations, we performed a luciferase, and again, you can see a noticeable reduction in the luminescent activity from the mutant biosensor compared to its the wild type counterpart. And right here, this is approximately a 20-fold decrease, which is a substantial drop in signal. Again, this decrease in luminescent activity wasn't due to varying amounts of protein lysate, again, shown using the Western blot. So now that I've established my biosensor, I'll now move on to the second aim, which is validating the biosensor both in living cells and in vitro. So the initial validation step is quite straightforward, determining if the luminescent signal is dependent on the amount of GSDMD intramolecular biosensor present. So to do this, cells were seeded into a 12-well plate, and duplicate samples were transfected with increasing amounts of plasmid, ranging from 0 to 0.6 micrograms. Following this, lysates were then extracted for a luciferase assay. And then from this luciferase assay, you can see a clear relationship between the signal intensity in the luciferase assays, as well as the biosensor expression level seen by the increasing band intensities from the Western blot, as well as, again, same amount of lysate for each sample. The next step in validation is determining if the biosensor can be stably expressed within cells, as the previous construct is limited to transient expression. So in other words, just the biosensor activity will be decreased over time with the previous model. Cloning was performed once again, and this time to insert the entire biosensor into the WPI vector. So this WPI vector is a type of lentiviral vector, which enables us to produce lentivirus. And this is capable of integrating the biosensor into the host genome for stable expression. The vector also contains green fluorescent protein, which is used to validate the successful infection. And the exact purpose of GFP will be become a little bit more clear in the upcoming slides. So to produce lentivirus, we started by introducing the WPI vector carrying the biosensor, along with packaging and envelope plasmids. So these other plasmids are just responsible for making the viral capsid, as well as the envelope surrounding it for integration. And these were transfected into the HEC293T cells. After allowing time for incubation, we collect and filter the medium containing the virus. And then this lentivirus, now carrying our biosensor, is then infected into MCF7 breast cancer cells, ensuring stable expression of the biosensor. We then use the green fluorescent protein to confirm successful infection, as its presence indicates that the plasmid has been successfully integrated into the cells and that the proteins on the plasmid are now being expressed. So the reason why we're using the MCF7 cell line is because they were found to have no GSDMD present, seen here on this Western showcasing GSDMD expression levels from a wide variety of breast cancer cell lines. And this was discovered previously by Derek. So that means that the cell line will only be expressing GSDMD that was integrated in, that being the GSDMD on the biosensor, rather than any endogenous GSDMD present. So you determine the success rate of infection using the GFP expression as an indicator, comparing locations of MCF7 cells under white light with green fluorescence under the blue light. So as you see here, this is the same slide the same slide of cells. So when you compare the locations here, you can see some GFP expression, although it's not the greatest. We really just need enough to have a substantial amount of luminescence activity when we conduct the luciferase assay. And we can see that that's actually the case here, where the MCF7 cell stably expressing the biosensor has approximately a 40-fold change compared to the control. Sorry, more like a 35-fold change, which is substantial enough for the project. So this stable line is what's going to be used for identifying small molecule inhibitors, and I'll cover this later in the third aim of my project. The next step in validation involves visualizing biosensor luminescence in living cells. So both wild-type MCF7 cells as well as the stable line were seeded into varying cell quantities in triplicate, ranging from 1,000 to 6,000 cells, as well as having 16,000 cells, and having the MCF7 cells as the control, having the highest amount of cells in the plate. So subsequently, luminescence detection was performed using the IVIS in vivo imaging system to evaluate signal intensity. The purpose of this validation is to not only show that this luminescence can be shown visually, but it also ensures that we can make a mouse xenograft model with these cells used to observe how small molecule inhibitors might disrupt GSDMD activity in a biological system. So from these results, you can see a nice incline in signal intensity correlating with the amount of cells plated, as well as no signal from the MCF7 cells without biosensor. So below, you can see the BLI analysis where we can see it visually, purple being low intensity and moving up to the red with the highest. And then this corresponds with luciferase assay. I might say a live cell luciferase assay using the same plate of cells directly after conducting the BLI. So transitioning into the final step of validation, I'll be discussing, I needed to characterize the biosensor within an in vitro model. This involves purifying a his-tag GSDMD intermolecular biosensor fusion protein. And the process begins by cloning this biosensor into the PET28B vector, which incorporates a polyhistidine tag, or affinity tag, and this attaches to the biosensor insert. So this vector was then subsequently transformed into the BL21 competent cells, commonly used for recombinant protein purification. And then bacterial colonies are then selected using cannomyosin-coated plates. Following this is the induction of protein expression using IPTG. And it's actually a pretty interesting system because then this regulates T7 RNA polymerase production within the cells, thereby then transcribing your fusion protein. Next, cells are lysed using sonification. And then his-tag fusion proteins are purified using agarose beads that selectively bind to this his-tag that we introduced. The proteins are then separated from the beads using size exclusion chromatography. And then finally, the proteins are subjected to an SDS page electrophoresis and stained with chromacy blue to quantify the protein amount. So varying amounts of fusion protein were loaded onto the gel, as you can see here on the right, as well as a BSA standard with known concentrations on the left. So when comparing these band intensities, 2 microliters of fusion protein aligns best with the 0.5 micrograms of BSA standard. And this was just chosen randomly to find the best fit between the two. And from this comparison, we can take a guess that the approximate protein concentration falls around 250 nanograms per microliter. So samples are then prepared using increasing amounts of fusion protein based on the concentration that we estimated. And then this underwent, once again, a luciferase assay. So sticking with the trend here, we do see a positive correlation between the luciferase assay or activity as well as the amount of fusion proteins loaded. So I also want to note too that it is quite a substantial increase in luminescence as we're getting up to 10 to the 6, as well as having relatively small amount of fusion protein, 80 nanograms being the highest loaded. So this just serves as a valuable tool for quantification of your fusion or purified fusion protein and validation when screening for drug inhibitors that influence GSDMD activity. So more specifically, it allows you to monitor GSDMD activity within an isolated system. If you introduce a drug inhibitor of GSDMD and your signal decreases, this is showing that your drug is directly interacting with GSDMD, again, in this isolated system. So one additional assay was conducted, adding increasing amounts of caspase-1 to the GSDMD fusion protein. And when caspase-1 was incrementally added, you can see here a clear dose-dependent decrease in biosensor signal. And this is just demonstrating that GSDMD-mediated cleavage of GSDMD can be observed in the in vitro system, just one step further characterizing this model. So now that the biosensor has been validated in living cells and in vitro, I'll move on to the third aim, which is identifying and investigating small molecule inhibitors that inactivate GSDMD using drug screens. So before getting into the drug screen, I'll provide an overview with the type of inhibition that I'll be investigating, specifically focusing on the phosphorylation of GSDMD as a post-translational modification. So GSDMD possesses several known phosphorylation sites, some of which have known functions and others remain unexplored. And since kinases are the proteins responsible for phosphorylation, my objective is to identify a drug that inhibits kinase activity, which is involved with the phosphorylation of GSDMD, thereby reducing GSDMD levels as a result of this inhibition, potentially through disrupting GSDMD stability. So here's a map displaying a collection of known post-translational modifications sites for GSDMD. And this was collected through a variety of proteomic screens, both high throughput and low throughput. So I also want to point out that the blue dots here are all the potential phosphorylation sites that they found. So by targeting kinases that in turn target GSDMD, we could disrupt the phosphorylation from happening at any one of these sites, phosphorylation sites, thereby disrupting GSDMD stability, reducing its levels within the cell. So based on this rationale, a kinome-wide drug screen was performed by the Lonenfeld-Tannenbaum Research Institute in Toronto, and they used a library of 780 compounds, 560 of them being known kinase inhibitors. On day one, the MCF7 stable cell line was plated into 386 weld plates. Then on day two, the kinase library, or the compound library, was then added alongside DMSO as controls. And then finally, on day three, luciferase activity was measured. So I'm only going to be showing one of the 6 plates that were read just to show how this data was processed. So as you can see here, you can see the raw luminescent data for one of the plates. And the mean signal was collected from the wells treated with the inhibitor. And also remember that each sample was done with 2 replicates. And this was then divided by the control sample, that being the MCF7 cells with the biosensor treated with DMSO. So then this gave us our mean inhibition of each compound, where positive hits had to be at least threefold inhibition or lower. And these are just indicated by the red text. So after doing just this from the raw luminescent data, we were left with 74 hits. The data was then further filtered using the Z-scores of each sample. So just to briefly explain it, the Z-score represents how far away the data point is from the mean of the data set. So in the context of our drug screen using a Z-score threshold of -2, this means that you're interested in identifying data points that fall more than 2 standard deviations below the mean of your data set. So each of these black dots here are labeled with the corresponding cell ID tag from each 364 well plate, representing positive hits after this filtration step. And after identifying samples that had Z-scores that were falling below this threshold from all the plates, we were now left with 21 hits. So following the screening results, we were then able to validate 19 of the 21 initial hits by completing the experiment, or completing the same experiment within our lab. So this involved diluting the inhibitors to 5 micromolar, and then treating with the MCF7 stable cell line with each compound in triplicate, as well as having several DMSO controls. So basically, the drugs were added to the cell line. Then 24 hours, we took the cell lysate, and then we conducted the luciferase activity to see the amount of inhibition these compounds are producing on the biosensor. So the outcome from the secondary validation is actually very encouraging, as you can see from the graph provided. And this is basically just showcasing the relative biosensor activity in comparison to the DMSO controls. So for this analysis, a cutoff of 30% biosensor activity relative to the control was established. So in other words, if the drug had caused at least 70% inhibition of the GSDMD biosensor. So from these results, 15 out of the 19 inhibitor-treated cells, which are highlighted with the red dots down below, met this criterion, leaving us just 4 false positives after the secondary validation, and having an overall success rate of 1.9% from the entire kinome-wide drug screen. I also want to point to, well, multiple of them did fall below the 30%. There is quite a few that are pretty potent inhibitors, at least seen here from the screen, having a relative activity of 0.1, so 10% of the activity of the biosensor control. In other words, having like around 90% inhibition. So the following table here shows all of the positive hits from the screen, as well as their respective kinase targets. So just working through the table here, they do show some pretty interesting results. So the first, the blue section up here shows inhibitors that target kinases that are involved with the PI3K, AKT, mTOR signaling pathway. And it's also another point I want to show is that there's multiple of the same kinases being targeted within this pathway. So it kind of shows that these different kinases it's likely that they're going to be directly targeting with GSDMD as well as, or rather than just having like a false positive trickle through the filtration process. Next is two that inhibit kinases involved with cell cycle regulation. And then there's also other kinases that don't fit into these groups, but they're also worth mentioning, such as the FAK inhibitor that targets focal adhesion kinase, or FAK for short, and the IMD 0354, which targets the inhibitory kappa B kinase, IKK beta. So now I'm just going to go over to the specifics of these inhibitors a bit more, or sorry, the kinases in which are being inhibited a little bit more regarding their involvement with cancer. So altogether, the screen has identified kinases targeting GSDMD that are also involved in a wide variety of cancer hallmarks. For instance, the PI3K pathway, which is a major signaling pathway involved with cell growth, proliferation, and survival, is commonly dysregulated in many cancers, being associated with increased cell proliferation, metastasis, and resistance to chemotherapy. Something that's worth noting is that this mTOR has actually been shown to be involved with pyroptosis already. So it's been shown to promote the production of reactive oxygen species within the mitochondria. And this in turn promotes the oligomerization of the GSDMD internals. So it's kind of interesting to see that from this screen, it's kind of pointing towards mTOR actually directly targeting GSDMD as well. Don't know how yet, but it's just an observation here. Next, CDC25 and the CDK4 cyclin D1 complex are key regulators within the cell cycle, specifically targeting at the G1 and S transition. So basically, the G1 and S transition is just it's where cells decide whether they want to proceed with cell division or they enter this resting state or the G0 phase. And also, dysregulation of these components can lead to uncontrolled cell growth, which is again found in cancer. Next is the FAK, which is a non-receptor tyrosine kinase. And this is and this plays a crucial role in cell adhesion. So if there's dysregulation within this kinase, this can lead to tumor cell migration and then metastasis with secondary tumor formation. So IKK beta is involved in the regulation of nuclear factor kappa B signaling pathway, again, where dysregulation led increased tumor cell invasion as well as metastasis. So the central question now is, how does GSDMD actually connect within these pathways, as well as how would GSDMD's role intersect with other cellular processes? Altogether, these findings from the Tynan-wide screen just deepens our understanding with GSDMD's involvement with tumorigenesis. They also provide an insight into the mechanistic connections beyond GSDMD's involvement in pyroptosis and cell mechanism, which were two processes that have already been well established. That being said, after the screen, there's still quite a bit of work to be done. So in the future, I aim to dive deeper into the implication of these findings, with the screen results effectively laying out the groundwork to further explore GSDMD in the context of these kinase inhibitors. And as of right now, this is basically the end of my results and where my project stands. So now I'll move into the future directions of my project. So next steps involve secondary validation steps for the kinase inhibitors. One will be a dose-dependent inhibition using these inhibitors to just gauge the IC50. I also want to utilize other types of biosensor, another type of biosensor called TR-FRET, which instead uses fluorescent signal as the bioluminescent signal found in nanobit. And this will ensure that the inhibitors identified aren't quenching the signal from nanobit alone, rather than instead inhibiting kinases that target GSDMD stability. Additionally, I aim to, gain a deeper understanding of the mode of inhibition exerted by each of these kinase inhibitors. So I'll conduct Western blot analyses to assess GSDMD intramolecular biosensor levels and how they change when kinase activity is inhibited. So what we think is going to happen is that, again, it's GSDMD stability is going to be decreased and there should be less GSDMD showing up on these Westerns. But for the off chance, maybe GSDMD cleavage might be occurring. So we might see a reduced band length on that Western. So just further characterize the mode of inhibition. Moving forward, I plan to conduct several cell-based assays, assessing how these kinase inhibitors affect cell phenotype. So for instance, kinase inhibitors will be used to suppress hypoxia-induced pyroptosis conditions found in heart failure. So hypoxia typically causes the accumulation of reactive oxygen species, and this is found to actually promote pyroptosis through the canonical pathway. Next is to see if these kinase inhibitors suppress LPS-induced pyroptosis, which is typically stimulated by gram-negative bacterial infections and causes that non-canonical route of pyroptosis to occur. And we're also going to use inhibitors to suppress or see if it suppresses cell growth within breast cancer cells that have high GSDMD expression. In vivo studies using mouse models will also be performed, providing valuable insights into the effectiveness of these inhibitors within a biological system. And lastly, the current kinome-wide screen kind of serves also as a proof of principle screen, as it was small scale in design. And this justifies large scale, high throughput screens utilizing this intramolecular biosensor, hopefully being able to identify a whole new set of drugs that can directly block GSDMD activity. One specific screen that we'll be doing is in the future is with a small molecule screen, which encompasses a 140,000 compound library. And this is lined up in collaboration with Emory University. So moving on to the final section, which is the summary significance. So after going through my project today, if there's any key takeaways to remember, let it be these 2 points here. So the first is I've developed a novel bioluminescent biosensor capable of detecting the activity and level of GSDMD with high precision and sensitivity. The biosensor stands out due to its versatility, enabling us to have real-time monitoring of GSDMD cleavage, degradation, and conformational changes, which offers a comprehensive understanding of GSDMD's dynamics within cellular environments. Next is that I've also successfully identified multiple kinase inhibitors capable of disrupting GSDMD activity. And these inhibitors hold immense promise as potential drug candidates for treating GSDMD-mediated pathologies, offering hope for the development of novel therapeutic interventions in various inflammatory diseases and cancer. So I just want to end off with a thank you to Dr. Yang for providing me with this opportunity to pursue this project, as well as his support and guidance throughout. Thank you to my committee members, Dr. Greer and Dr. Jia, for their advice and guidance in navigating me through the project. I also want to thank all the collaborators that have contributed to the project, contributing their expertise and hard work, as well as a final thank you to all the members of Yang Lab for their everyday support and guidance. Thank you.
I'm sure Tynan would love to answer questions now. So folks in the Zoom, if you can either go to the chat or just chime in and we'll keep an eye on you on the screen. Questions? So have you tried protease inhibitors?
Protease inhibitors?
Yeah.
No, not currently.
Okay. But you used caspase in vitro. I think I sort of looked away for a second.
Yeah.
You killed the activity with the caspase, which is expected.
Yeah. So you're basically saying to inhibit just caspase activity?
Yeah, or other proteases that might be involved, but you certainly have candidates for sure that you know are involved.
Yeah, that's like one aspect of it. But so with the protease inhibitors, I assume you're getting at the caspases that cleave GSDMD, right?
Yeah.
As well as maybe others.
Yeah.
But that cleavage is only one aspect. So going back to that like two-pronged approach in GSDMD and different types of diseases, we don't think that GSDMD cleavage is part of the tumorigenesis or tumorigenic process. Instead, it's actually having alternative mechanisms, like Derek found the metabolism or the binding partners, but also other pathways that I talked about too that haven't yet fully been explored.
Well, that's something that maybe you can expand on a little bit. The idea that you have certain functions that are dependent on pyroptosis and others that aren't. So can you talk a little bit more about that? So yeah, pyroptosis and release of inflammatory cytokines, but you don't think that that's that could contribute to tumorigenesis?
Well, I guess in a way, and they've like kind of hinted at it in the literature, where pyroptosis, having pyroptosis would actually be good for to like elicit an immune response against the tumor. But they found that an effective GSDMD has been found in multiple of these cancers. So you're getting an anti-tumor immunity here.
On top of the protein? Yeah. Okay. Yeah. I got lots of questions. Okay, I'm gonna go again. So you touched on, did you get any hits in your screen that actually increased signal instead of you were looking for things to decrease?
We did actually.
Yeah, I thought I noticed some.
Yeah, I didn't really touch on the ones above, but from the Z-score, there's actually some that hit above. So
any idea what's going on with those?
Actually increasing biosensor activity. It's a good question, actually. Maybe it's actually promoting some sort of GSDMD stability. So my direction I'm going is that the lack of phosphorylation actually decreases it. But maybe in this case, these kinases are being inhibited that actually promote. So maybe it's like just more a dynamic process of GSDMD. And there's also multiple sites too. And they found so the phosphorylation sites of GSDMD, they found the ones that they've investigated, they primarily are found on the N terminal and have to do with that oligomerization. Yeah. So they find that they're actually being phosphorylated, and then the phosphorylated versus the non-phosphorylated actually have promoted oligomerization after it's being cleaved.
So you used your biosensor and did some nice structure function looking at those points of interaction between C-terminal and N-terminal domain.
Yeah,
that looked very impressive.
Yeah.
Have you or do you plan to make phosphomimemic or non-phosphorylatable type mutations where many of those phosphorylation sites are? So
yeah. So that's actually one of the next steps in validation. Okay. So the first thing that we're going to do is we're going to use the in vitro system and then determine if it's being phosphorylated or not. But then we can start actually integrating these mutations into it. First, what we can do is we can actually break up the protein, like using proteolysis, and have these small fragments. And then once we find the segment in which there's potential phosphorylation sites, we can then introduce mutations there.
Yeah, but I'm assuming that you don't need to map all of them, because that screenshot you showed was phosphosite website, and so those are sites that have been determined just in mud pit phosphoproteomics to be phosphorylated. So you've got a whole bunch of candidates that you could start with without having to. You're talking about trying to map phosphorylation sites yourself on. Yeah.
So one thing is actually making sure that they are phosphorylating GSDMD, and another is where. So first, figure that out and search that.
Okay. Question over there.
Does it seem like any of those kinase classes would be specific enough to just phosphorylating GSMD, or would they, by inhibiting them, you would inhibit a lot of other kinase functions, and you would run into the same problem that the other current inhibitors with off-target?
Yeah, that's another good question too. And I guess that's just, I'll start off by saying there are known kinase inhibitors using for drug therapeutics. Like there's Gleevec that inhibits the kinase responsible for chronic myeloid leukemia. But of course, these kinases have a bunch of different targets here. So if you're inhibiting these kinases directly, maybe other pathways are being disrupted. And I think that's when you start to get into that. You try to find the effective dosage that would inhibit GSDMD specific pathologies versus then you start getting that cytotoxic effect. But you're right. Like it's something to keep in mind too when we're getting to later stages and we have a developed inhibitor as a potential drug therapeutic. Yeah.
So you might want to bring up the chat yourself so you can. Yvonne has some.
Can I just read it through?
Yeah. Yes. So I have two questions. First question: can you be sure that you are seeing Gasdermin specific cleavage in your caspase assay, and not simply cleavage of the nanobit away from Gasdermin, or a combination of the two? So that's the first part of that first question.
Okay, so when we're,
Did you look at the protein after the caspase cleavage?
that's actually something that we're currently working on right now. So that's actually very interesting because that's kind of one of the roadblocks I'm on right now. See, when caspase mediated cleavage occurs on GSDMD, it causes the N-terminal to oligomerize and pyroptosis to form. So when we're collecting these cell lysates, all the cells attached to the cell plate right now presumably doesn't have that cleaved GSDMD because they're still alive. Pyroptosis hasn't occurred.
Mhm.
So something that we need to look into is actually taking the the media containing the dead cells on top and then seeing if there's a reduced band length there to
Okay.
show that these caspases are
Yeah, and you could try that in vitro too, right?
yeah, exactly. Yeah.
Okay, and then the second part of the first question is not that this is material to your high throughput screen, but that could be important in terms of biosensor validation alone. Okay, that was just a comment. Yeah, just validating how the biosensor is working. So question two, is there strong benefit or detriments to going after Gasdermin versus going after its kinases?
I mean, I feel like the best form of inhibition is targeting GSDMD directly. Like,
You're biased.
pardon? Yeah.
It's just that there's all those pretty kinase inhibitors all
Yeah.
over.
Yeah.
Okay.
I mean, this is just one avenue we're initially going for, and mainly it's just logistics. We had this kinase screen lined up. But of course, if we can find a small molecule inhibitor that targets GSDMD, and this is kind of more so what we're doing with that high throughput screen, that probably or ultimately would be a better option because it's more of a directed therapy towards GSDMD.
Because a lot of those.
I know I keep saying GSDMD directly, but I guess technically the kinase inhibitors isn't, but in turn the kinase should be directly targeting GSDMD.
And I noticed that one of your hits was storosporin.
Yeah.
So that speaks to the lack of specificity of kinase.
Yeah, that one's the pan kinase.
Yeah.
Right? Yeah.
Yeah.
That can use more like a positive control too.
Yeah. So congratulations on a chunk of very elegant technical work with nice data.
Yeah.
But I mean, the biology, I think, poses incredible questions. What you have already said, these are
Yeah.
6 pages of questions there for you. You've got these processes of pyroptosis, which in itself is interesting, and then tumorigenesis. And so, I mean, a question that comes to me is, so what does this protein do functionally, physiologically, let alone pathophysiologically? And it looks as though you need to activate
Yes.
GSDMD.
Yeah.
So you either have to have some conformational chain or cleavage.
Yes. Yeah.
That's right. So the pathology here is something. So you've taken this nice hint of using biosensors to look at things that influence pathways, which may involve tumorigenesis. So I think your future study slide.
Yeah.
You should put that up because I think that's about 6 PhDs worth of clinical needs.
I'll be here for a while.
If you get through one of them, I would say you're doing well.
Yeah.
I mean, it's great to be ambitious, but
Yeah.
but I think that is
See, yeah.
something.
Yeah. Like the blunt of the work has been actually producing this biosensor. I don't know if you remember from my first talk, my original biosensor was determining between GSDMD and caspase-1, but we just couldn't get it up and running. Like it was shown to be working in vivo, but not in vitro. So that whole segment took like 8 months, and now we're on here. So now we're just like,
Plan A, B, C.
yeah. So the now that we have the biosensor working, it kind of opened up in the kind of wide screen. It's like opening up a can of worms. Like it's just so many different possibilities now.
Element that sensitive system.
Yeah.
And if you can answer some of the biology,
Yeah.
which I think is critical and fascinating, but
Yeah.
it's really unclear to most of us how these things
Yeah.
come together.
Yeah.
And what is the physiologic, let alone the pathophysiologic function of this protein?
Yeah. Like the kind of wide screen, that's also too why we did it, because mechanistically it now shows how GSDMD plays into these other pathways. So if we can start integrating it into other cellular processes, as well as inhibiting, it'll just give further insights into what this protein is doing. Because really, we don't know what's happening right now in the grand scheme of things. Planning to. So that's what I, yeah. Yeah.
So, I'm not finished on my question. We have 7 minutes left by my counting.
All right,
So
your way.
I noticed that you're planning on going down to Emory and screening a big library of small molecules, so
Yeah.
that's pretty cool, and good luck with that. That's not my question.
Thank
So
you.
when you looked at the phospho site, PTMs and the highest one up there was a phosphorylation site.
Yeah,
So, did it give any, was it just, hits in big screens, or were there any publications that alluded to functional roles or
Yeah.
kinases responsible, that sort of thing?
that one that had a lot of different hits from the proteomic screen. I forget what the number is, but it's a serine residue. And they actually found kinase AMPK to phosphorylate that. And that phosphorylation actually goes back to, I think most of the phosphorylation has to do with this oligomerization.
Okay, so there's already some known functional
Yeah.
relevance to that phosphorylation site.
Which is weird because when I was
Did
reading
was there a PNK,
AMPK,
MPK inhibitor in your hits?
no, I don't know.
Uh,
Something there. Yeah,
Hands-on one.
Yeah.
Yeah,
yeah.
good. Maybe there would have been.
There is also, I forget the actual outcome from the paper, but they did find an inhibitor of the AMPK. So actually, sorry, let me backtrack a little bit. The AMPK phosphorylates GSDMD, which promotes it. So then having the inhibitor that inhibits AMPK.
Yeah, you wouldn't see
Yeah,
it.
yeah. Some of the process what we're doing for the drug screen.
Yeah, so that gets.
So that's why that big
Well,
band is popped
and-
up there too with the
The
serum.
question that I asked about getting hits that actually increased signal is a little bit counterintuitive that you get that.
Yeah.
Because you've already got it in the confirmation
Yeah,
where you're gonna get signal.
Yeah.
And so, anything that increased that signal would presumably just cause some kind of conformational change that better align small and large
Yeah.
to some extent. So you're less likely to find those things relative to things that
Yeah.
break it apart.
maybe
Yeah.
too, like in the future as well with those upregulated GSDMD, maybe for tumorigenesis to make it maybe like a cold tumor to a hot tumor sort of thing with I don't know, promote GSDMD.
Sure.
Because they've also seen too that there's a lot of polymorphisms with GSDMD, having the single nucleotide polymorphisms, not within the phosphorylation sites, but they've also shown that these polymorphisms changed the stability of GSDMD.
Well, that raises lots of possibilities then, additional
Yeah.
possibilities going into data sets and trying like the UK Biobank and looking at whether there are differences in survival rates or cancer incidence in people with specific SNPs in the gene.
Yeah.
So, cool. I don't see anything more on the Zoom crowd. And no hands. Oh, was that a hand up? No, you just. Oh, there's, go ahead, there you go.
Question for you, when you showed the graph of all the phosphorylation sites.
Yeah.
What's the phosphorylation site in the way as we start to think about that being.
Yeah. So the acetylation is actually another regulatory mechanism for GSDMD oligomerization. So when it's acetylated at that group, it actually inhibits the oligomerization from happening. So I assume that it has some sort of conformational change within the C terminal to allow it to be released from the N terminal. But yeah.
All right, well, thank you very much. That was lots of fun.
Well, it brings up questions too that I need to now further explore. Helps me.